Hi, this is Dr. Kim Pesca-Saludo again. So we're going on to this next part of the lab, which is going to be talking about cell cycle and mitosis. As you can see, we got this from chapter 4 in the book. We should already have learned about all the organelles in the cell last time in lab, and today we're going to be talking about cell division as well as the cell cycle. And so there are a couple of different types of cell division that we'll talk about in these two courses, so Bio 168 and 169. And we've split them up a little bit so that we're talking about mitosis in Bio 168 at the very beginning, just as part of the cell cycle. And then we're going to wait a little while and talk about meiosis, and then we'll compare meiosis with mitosis again and do a review of mitosis in 169. So now what do those words mean? Meiosis is going to be this type of cell division that occurs only in sex cells or in gametes. So those cells that eventually will turn into sperm or eggs. And so that's going to start with a diploid number of chromosomes, which is 23 pairs. So we have 2 pairs of each, and you'll end up with a haploid number of chromosomes. So 23 chromosomes that are unpaired. And we're going to cover that in Bio 169. But mitosis is the type of cell division that occurs in somatic cells. And that means that like all of your body cells, all the cells that are not sex cells. And in somatic cell division or mitosis, one cell divides to produce 2 cells. And these cells are diploid cells. That means they have two sets of chromosomes. So you'll have one from your mom and one from your dad. So you have 23 pairs of chromosomes. So 2 sets of chromosome 1, 2 sets of chromosome 2, 2 sets of chromosome 3, and so on. So your body cells start with 23 pairs of chromosomes, then they replicate the chromosomes, and then they divide. And so you end up with 2 daughter cells that each have 23 pairs of chromosomes. So they're should be exactly the same as the original cell. This is necessary for development, for tissue growth, replacement of old cells. You know, if you scrape your knee, right, or you scrape your arm, you're going to need to replace those tissue cells. So I want to review one of the structures that we talked about last lab, and this is the centrosome. And remember that a centrosome is made up of a pair of cylindrical centrioles. And so these are in charge of like managing the body's or the cell's cytoskeleton, so those microtubules. And these microtubules are going to be really important in moving the chromosomes around and forming the mitotic spindle. So the centrosome is kind of the organelle that's going to help control cell division. Other things that will be important of are DNA that's in the cell. So remember that the DNA is found in the cell within the nucleus. And normally the DNA is in this sort of loose, uncoiled form called chromatin. And we identified that on our model last week. It was kind of that pink fuzzy stuff inside the nucleus. And remember we said that our nucleus normally in our somatic cells is diploid, so it has 23 pairs of chromosomes. So 23 times 2 is 46. So we'll have 46 chromosomes. And as I said, normally when the cell is just, going about its normal business, they're loosely coiled. When they're loosely coiled as chromatin, things like transcription and translation can happen and all of that. Once they become tightly coiled in chromosomes, they are not as all of the different enzymes can't really get to them, and we can't make proteins, and we can't use them as much as blueprints for the cell. So these chromosomes, they don't become tightly coiled as chromosomes very often. It's really only just during cell division. And so when they become tightly coiled chromosomes, that's going to be during the mitosis phases. And we'll talk about like when that happens. So let's kind of move back a little bit and talk about the cell cycle. And the cell cycle is going to be an important sort of phase that each cell is going to go through. And as the cell goes through these phases, it's going to have to go through different steps. And these steps are going to be important. They have to go through them in order to replicate that somatic cell. Once it goes through the cell cycle and kind of completes that whole cycle, it will divide into 2 identical cells, and we call those the daughter cells. There are 2 major phases in the cell cycle, and they're called interphase, and that lasts approximately 23 hours, and the mitotic or M phase, which lasts about 1 hour. So I think about it's like a day, right? So it's 24 hours total, and one of those is for mitosis, and the other 23 are for interphase. And so this is kind of our the picture of the cell cycle. where we have interphase for most of it. This is not quite to scale, right? Because it looks more like a quarter instead of like 1/24. So it's not quite to scale, but know that interphase is a lot bigger. And it has its different subphases, which we'll go over in just a few minutes. And then mitosis, and it's followed by cytokinesis. So we're going to go through each of these different phases in just a moment. But you might want to come back to this picture and it'll be good. Make sure that you can label this picture and kind of the different phases and what happens in each one. So interphase, as we said, is going to be this main blue area here. So this whole thing is interphase. It lasts around 23 hours. And this is while the cell is preparing for cell division. And in this time, the DNA is loosely coiled as chromatin, right? Because we're going to be replicating the DNA, and it needs to be loosely coiled in order for our nucleotides to go and bind, and for our DNA polymerase to be working. And so there are 3 phases in interphase: G1, or GAP1 phase, S, or synthesis phase, and G2, or GAP2 phase. And so the G1 phase is going to be first, and this is when we have growth and production of new organelles, because we're going to be taking one cell and splitting it into 2 cells, so we need double the amount of organelles that it had before. We also need to kind of grow its cytoplasms. The structures needed for DNA replication are formed too, because at this point the cell has 1 copy of all the DNA, but we need to take one cell and divide it into 2, so we need to make another copy of that DNA, and so we need structures for DNA are going to be formed. And then finally, we have the replication of the centrioles to produce 2 pairs of centrioles at the end of G1 phase. And replication of the centriole. So that now we have 2 pairs of centrioles, which means 2 centrosomes. After that, we have the S phase. And the S phase is important because this is where DNA is replicated. And so it's loosely coiled right now, and we're going to be starting with 23 pairs of chromosomes, and so it's 46 total, and we're going to replicate each of these chromosomes. So we start with 46 single chromosomes, and we're going to end up with 46 replicated chromosomes. And this requires a couple of things. So deoxyribonucleotides. So remember, we talked about the nucleotide bases in chapter 2, our biological macromolecules. So for DNA, we have the bases A, T, C, and G. So we need those bases in order to make another copy, in order to those are the monomers in order to put them together as a polymer of DNA. And we need DNA polymerase, as well as a bunch of other things, but these are the main pieces. All are going to be found in the nucleoplasm of the nucleus, and that's like the cytoplasm inside the nucleus. So I want to stop here and talk a little bit about DNA replication. Now, if you've taken a biology course or a cell biology course before, you've probably gone into this in a lot more detail. We're going to kind of strip it down and go over the basics of DNA replication. And if you guys have learned about it before, as I said, this is going to seem a little bit simplistic. But for this course, we're just going to talk about the basics. So there's a couple of steps, and we're going to talk about, make sure that first you know that your DNA is going to be in the loosely coiled form. So it's in chromosome. And so since it's in chromatin, since it's loosely coiled in chromatin, we're going to unwind it. We're able to access it, and our polymerase can access it. So we unwind the DNA molecule. So it's in a double helix, and we unwind it and break apart the parent strands. At this time, we have new DNA strands are then going to be assembled. So those base pairs are going to, complementary base pairing is going to happen. I'm going to talk more about this when we have the picture, it'll help. And then the DNA double helix are going to be restored. So let's look right here. So first we have our DNA right here. So it's wound up in this twisted ladder or double helix form. So first we're going to unwind it. And so we're going to straighten it out a little bit. And as we unwind it, so this is right here, this section's unwinding. And then we're going to split these 2 strands apart. And if you'll notice, it says right here that we're breaking hydrogen bonds between the bases of the 2 DNA strands. So normally we have A bound to T and C bound to G, and they're each going to be bound by hydrogen bonds. And these hydrogen bonds, remember, are sort of weak-ish by themselves, but a lot of them together are strong, and they're going to be bonding between a partially positive hydrogen and a partially negative something else of polar molecules. So we have hydrogen bonds here, and they're going to be broken. So when we break these hydrogen bonds, we can actually split these 2 these two strands apart. So we're going to break them apart. Now we have half of one strand on this side and half of one strand on the other side. And so at this point, remember we said we need to, eventually we start with one strand and get two strands. And so what we're going to do is take each of these half strands and we're going to use that as a template in order to make a second, in order to make a new strand. So we're going to take these DNA deoxyribonucleotides, so these little nucleotides are A's or T's or C's and or G's, and DNA polymerase is going to go ahead and bind them on. And so they're going to bind them on in the proper order. So if you have an A, you know it binds to a T. If you have a T, you know it binds to an A. If you have a C, you know it binds to a G, and so on. And so all of this is going to be happening as our DNA is unwinding. It's going to be happening on either side. Now we have a leading strand and a lagging strand. One of them is going to go it's going to be straight and uninterrupted. The other one is going to have our Okazaki fragments. We're not really going to go into that much detail here. So I really just want you guys to know that we're going to split them apart. This is why we call it semi-conservative, because we're conserving half of the original strand of DNA. So these new strands of DNA each have half of them are made of the original strand of DNA. So it's semi-conservative. And then DNA polymerase is going to bind these nucleotides here to make a new strand. And they know which nucleotides to bind because A only binds to T and C only binds to G. And so this process is going to continue until both strands are replicated. And these replicated strands are going to be attached in the center at something called a centromere. So they don't come apart, so they actually stay together. So we originally had one chromosome, and now we have a replicated chromosome that's attached together at the centromere. And these replicated strands, we call them sister chromatids. So the two new cells are daughter cells, and these replicated strands are called sister chromatids. There we go, this way. All right, so DNA replication, if we have time at the end, I will direct you to, or I'll probably add a nice link for practicing DNA replication, because a video does that better than my trying to explain it based on a picture. But so after our S phase, so remember, an important thing to know is that the S phase is where DNA replication occurs. So in the S phase of interphase. Now, next we have the G2 phase, or the gap2 phase. This one is a brief phase that happens after the S phase, but before the M phase. And it's where our centriole replication is completed. And then some of the enzymes we need for cell division are synthesized. Once the G2 phase happens, then we're going to be moving on to mitosis. But I do want to mention something, and these are called the cell cycle checkpoints. And there are a number of checkpoints within the cell cycle to make sure that the cell is actually ready to undergo mitosis. The three most important ones that we'll mention are going to be the G1 checkpoint, the G2 checkpoint, and the spindle checkpoint. And so the G1 checkpoint happens between G1 and S, so between the G1 phase and that synthesis phase. It checks for adequate cell size. So if the cell is not big enough, it's not going to be able to divide very well. So it needs to get big enough so that each of our, it has enough cytoplasm and organelles and things to be able to distribute them to two new cells. It needs to make sure it has enough nutrients for that one. And it also needs to make sure that the DNA in the cell is healthy, because if we have DNA that is not healthy, we don't really want to go ahead and replicate that cell. And so this is the most important checkpoint, and usually if it passes this one, it's going to pretty much undergo mitosis. If it doesn't pass this one, it might go back into a resting phase called a G0 phase, and that's kind of like a resting phase until it, builds up enough, adequate size or gets more nutrients and things. The next one is the G2 checkpoint, and that is going to happen between G2 and M. So right where we talked, like right now we just talked about G2 and we're moving into M. So it happens in between interphase and the mitotic phase. This is going to check for DNA damage and DNA replication. So remember, by G2, our DNA should already have been replicated. So we want to make sure that we have enough DNA to, distribute to both of the cells and to make sure that the DNA was copied correctly. So the cell is going to double check that, make sure that the DNA is working properly and it's copied correctly. And if it finds anything wrong, it can pause and fix that, and before it moves into mitosis. And then finally, the spindle checkpoint happens between metaphase and anaphase in mitosis, and we'll start talking about what those phases are in a little bit. And it checks for whether all the sister chromatids are correctly attached to the spindle microtubules. And so we'll talk about how that, like, where that happens once we get there. All right, so this is a picture that kind of shows the S phase. This is interphase, G2 phase, mitosis, G1 phase. So this is a little bit rotated from our other picture, and I got it from this Khan Academy article, and you can read more there. And so let's see, G1 phase here. At the end, between G1 and S phase, we have this G1 checkpoint. If it passes the G1 checkpoint, it's going to go on to S phase. If not, it might need to stay in this G0 or resting phase and kind of get bigger and get ready for mitosis, wait for conditions to be better. Then next, like say if it did get to the S phase and it's going to go to the G2 phase, then we'll do the G2 checkpoint. That's going to check and make sure the DNA was correctly replicated in the S phase, as well as making sure that there's no problems there. After this, we'll move into mitosis. And the spindle checkpoint happens in between metaphase and anaphase to make sure that all the spindles are properly lined up and none of the chromosomes are like lost and floating somewhere. Each chromosome should be attached to a spindle microtubule. So now I'm going to move into, let's say that we went through all these, the G2 checkpoint, and we're going to go into mitosis. And so the way to remember the phases or the order of the phases of mitosis is PMAT. So the mitotic phase consists of these 4 phases: prophase, metaphase, anaphase, and telophase. So PMAT. So that's the mnemonic. And mitosis is going to be the division of the nucleus. So this is all talking about what happens in the nucleus. Whereas there's another process called cytokinesis, which is the division of the cytoplasm and the organelles. And so they happen, usually mitosis starts earlier, and then cytokinesis is going to start happening at the end of anaphase, the beginning of telophase, and it's going to continue, at the end of telophase. Until when mitosis and cytokinesis are both done, we'll have 2 different cells. So prophase is the first stage of mitosis. It's the first stage here. And this is where the chromatin, which was loosely coiled in interphase, is going to become supercoiled into chromosomes. And when it does this, it's going to turn into this little X look right here. And so you can see how the yellow part here is a centromere. And this is a replicated chromosome. Whenever it looks like this, it's an X, it means it's already been replicated. So it has 2 different, so this is like half of the DNA on the left side, and the other side might be the other half of the DNA. So we have our replicated DNA. So this is going to be 2 copies of that chromosome. So at this point, when it's supercoiled, we should have 23 pairs or 46 total of replicated chromosomes. And so these right now, we'll start seeing these chromosomes condense, and it'll look darker here on our nucleus. The chromosomes, you can start seeing them here. At the beginning of prophase, we're going to have this, our nuclear envelope is going to start breaking down. And if we had a nucleolus in the cell, it's going to start breaking down. Now our centrioles here, the centrosomes have moved to the opposite ends of the cell. So they were together when at G2 they had finished replicating, and they move along to the opposite ends of the cell. So one is on each side, and they start sprouting these little microtubules. And these are the spindle fibers. And these spindle fibers are going to grow from the centrioles towards the centromeres of each of these chromosomes. And so at this point in prophase, everything's still sort of organizing. And we have our developing spindle here. The nuclear envelope is disappearing. So by the time the nuclear envelope is totally dissolved, that's going to mark the end of prophase. Prophase is a pretty long phase. And in sometimes, some books, it might divide prophase into like an early and late prophase, or they might call it, at the end of prophase can sometimes be called prometaphase. So if you've learned that before, that's kind of like the end or late prophase. So next is metaphase. And metaphase is going to be the second stage of mitosis. And at this point, all of the chromosomes are lined up at the center of the cell. And you can see in this picture right here, they're in a really pretty line. And we call this the equatorial plate or the metaphase plate, where they're all lined up like this. The spindle fibers that have grown from these centrosomes should be attaching to the chromosomes at that centromere. And at the end of this part, in between metaphase and anaphase, that's where we have the spindle checkpoint to make sure that each of these chromosomes is going to have a spindle from each of these centromeres coming, or from each of these centrosomes attaching to the centromere. And so each of these replicated chromosomes is going to be attached. So one half of the centromere is going to be attached to one centrosome. The other half is going to be attached to the other centrosome by these spindle fibers. And all of these spindle fibers together, we're going to call the mitotic spindle, and they're made of microtubules. So whenever you see all of these chromosomes lined up right in the middle, think metaphase, right? That's the equatorial plate or the metaphase plate. The next phase is called anaphase. And anaphase, I think anaphase is going to be, as long as it passes that spindle checkpoint, all the chromosomes are attached, it's going to move on to anaphase, and it's going to start pulling those centromeres apart. So anaphase is apart. The sister chromatids are going to separate and move away from each other. And it's going to start as the spindle fibers move the sister chromatids apart towards the poles. So these microtubules start shortening and drawing these sister chromatids towards the opposite sides of the cell. So now those replicated chromosomes have now been separated. So before we had 46 replicated chromosomes, and now we've split the replicated chromosomes apart. Each new cell just has 46 chromosomes. So they still each have 23 pairs of chromosomes, but the big cell after the S phase, that at the beginning in prophase, it had 46 or 23 pairs of replicated chromosomes. Now we've broken the replicated chromosomes apart, and each new cell just has 23 pairs of chromosomes. So they were diploid at the beginning, and they're diploid now. So they still have 2 pairs of chromosomes, one from mom and one from dad. And so this is a good picture of an actual cell. We can see that these sister chromatids are now moving apart and being pulled apart to opposite sides of the cell. The other thing we can see here is this little guy here is called the cleavage furrow. This is the beginning of cytokinesis. So this little, where it looks like the cell is like tightening its belt, that's the cleavage furrow. And usually it starts at the end of anaphase and it's going to move through telophase. So telophase happens when the chromosomes arrive at each pole. They're going to then begin to uncoil and form chromatin again. And a new nucleolus is formed and a new nuclear envelope is formed. Once the chromosomes arrive at each pole, we don't need to pull them anymore. So the spindle fibers are going to kind of start breaking up and dissolving. The microtubules are not going to need to be connected anymore. So these are going to be moving through and our new nuclear envelope is forming. So once we have the new nuclear envelope formed, I think of that as like our telophase. And that's the end of nuclear division. But see, at this point, if the cell, usually at this point, we still have a little bit of cytokinesis to go through. So we still haven't divided the whole cell apart, even if we've divided most of the nucleus. So even if mitosis has ended, we still need to complete cytokinesis. So this is, again, still the cleavage furrow of cytokinesis. So cytokinesis is then the division of the cytoplasm and the organelles between those two newly formed cells. Well, the cytoplasm includes the organelles. It's going to overlap usually with anaphase and telophase. And so at the the outside, in the plasma membrane, there are rings of microfilaments. So we had microtubules in the spindle fiber. Those are the biggest ones. And then microfilaments are going to be on the outside, making up this cleavage furrow. And they're the smallest ones. They are going to pinch the cell into 2 separate cells. They're kind of contractile proteins. Think actin, which is contractile. We'll learn about that in muscle cells. It's going to kind of contract and pinch in and cause the mother cell to divide into 2 separate daughter cells. And after cytokinesis, the cell division is complete. So I really quick wanted to mention a little bit about tumors. And tumors are usually basically uncontrolled cell division. So the cell is dividing more than it should be. And a lot of times this, the regulatory mechanisms, things that normally signal the cells to divide or stop dividing, don't work as well. So some sort of cell signaling is disrupted. And so a cell that normally, like the DNA is damaged and it would say, hey, stop dividing, you shouldn't divide, for some reason it's going to keep dividing. And so if those regulatory mechanisms are not working properly, then the cell is just going to keep dividing. And so this uncontrolled cell division is going to just lead to overgrowth of that type of tissue or tumors. And this can interfere with the function of the normal cells surrounding it. These tumor cells can also enter the blood or lymph and move or metastasize to other areas of the body. Now, I did want to talk a little bit about, after the cell has divided, how cell aging and death. And so we did say that if the cell doesn't pass these checkpoints, and we go back up to the checkpoints here, like sometimes it can't pass these checkpoints here. Let's say that the DNA was like damaged during the S phase, somehow it didn't replicate properly and it couldn't fix it. If that happens, then usually the cell is going to be like, well, I don't want to, you know, take these mutations into the future. So I don't want to continue to replicate if I have these mutations that I can't fix. And so it's going to undergo what's called apoptosis, or programmed cell death. And so we'll talk a little bit about that in a second. And so I'll talk about apoptosis first, then we'll come back to aging. So apoptosis, if the cell decides that, you know, it is too damaged to continue, or if it's, you know, infected by a virus and it, you know, it needs to undergo cell death, you know, for the better of the rest of the organism, it's something that occurs in an orderly process. It has lots of steps that happen. It's going to destroy and remove the cellular components and the cell remnants. And it's usually initiated by ligand receptor signaling. And that means that, like, something tells the cell to do this. You have a chemical that binds to a receptor that says, hey, you should undergo apoptosis. And so there's a bunch of different enzymes that are going to happen during apoptosis that are going to be involved. I'm not going to ask very much about the details of it, but I want you guys to know that apoptosis is a type of cell death that is non-inflammatory. So it doesn't cause an inflammatory mechanism. And it can actually be healthy, part of normal development. So first, things that are going to happen, we're going to destroy DNA polymerase. It digests the DNA. Then the digestion of the cytoskeleton is going to happen. The cytosol kind of condenses and all the organelles get destroyed. And the cell kind of just shrivels up. And there's a picture, I think, in your book, but I don't think it made it here. And so it kind of shrivels up, but it doesn't, if it's filled with viruses, it's better about not releasing them as much, and it also doesn't usually cause as much inflammation. So programmed cell death can occur in proper development. So like when you're first, you know, a baby, an embryo, you, instead of having fingers like this, you have more of a paddle, right? And so the tissue in between your fingers underwent apoptosis in order to like give you separate fingers instead of having like a fish-like fin. So that can be important. So for proper development, apoptosis can happen. And also to remove harmful cells. So cells that may be cancerous, right? We said if they didn't, those checkpoints that, if they don't pass those checkpoints because there's like mutations and the cell's cancerous, sometimes it'll undergo apoptosis. If the cell is infected by a virus, sometimes it'll undergo apoptosis so that it doesn't spread that virus, things like that. So I said I'd come back to cell aging. So cell aging is kind of normal. It's a continuous process, and most cells are going to reduce function. So they're not going to be able to process all those reactions as well, and they're not going to be able to maintain homeostasis as well. As you get older, people are going to usually have a lower number of normally functioning cells, and then some of the cells that remain are going to have abnormal functions. They're not going to be able to work as well. So these cells usually have the structures of the organelles might alter. They might be different. They might not have as many organelles. The organelles might not work as well. And then also, as you age, you're going to accumulate over time changes in the structure of the chromatin and the chromosome. So your DNA can have changes in it. And this is usually why you see tumors and cancer arising a little bit more often in people who are a little older, just because it is more likely that you have a buildup, like usually cancer, you need a couple mutations in order to get a tumor or cancer. And so the older you are, the more likely it is that you've had more of those mutations. So that's why it's more a disease in older people. But you can definitely also see it in younger people, especially if they've been exposed to any carcinogens or may have a genetic predisposition. And so I said cell death can happen two ways. Once the cell can be killed by harmful agents or mechanical damage, like you cut the cell in half and it dies, or once again, they can program cell death called apoptosis can happen. All right, so that is going to be the end of our lecture. Please remember to look at the rest of your lab folder and go through the other parts of this. There is a really good virtual microscope portion of this lab. So make sure you go through and look at the virtual microscope and look at the onion root tip mitosis and the whitefish blastula mitosis slide, so you can identify those different mitotic figures under the microscope. I'm also going to add a link to more information about the DNA replication. So I'll add a couple links there so you can have some interactive DNA replication things to play with, because it's a little easier to see with the interactives in the videos than it is here. All right, thank you.